So with certain aspects of meiosis, here we have, and of course there's two phases of meiosis, meiosis 1 and meiosis 2. Now we'll get to the differences in those processes in a minute. Now here we have the depiction of our synaptonemal complex. And what the synaptonemal complex is a system of proteins that basically end up connecting the two homologs, the two homologous chromosomes, as you can see here. It connects the two. Okay? But when we actually draw it out physically, you see two essentially overlapping X's versus having them completely on top of each other, because that wouldn't make too much sense, since it'll just simply look like the same X. But essentially, this is what happens. You have the very close association, basically on top of each other, of the chromosomes, of the homologous chromosomes, via the use of the synaptonemal complex, which is just a structural protein complex that links the homologs together. So, let's see. One of the most important processes that occur during meiosis 1 is something called crossing over. And so crossing over, as you can see up here, we essentially literally have a cut and paste mechanism during prophase 1, where the close association of these homologs allows for certain sites to actually be combined, and they literally switch from one arm to another, as you can see right up here. You cross over from one arm to the other, from one homolog to the other, which allows for genetic recombination, and that's very important as far as maintaining genetic variability within the population. And with genetic variability being maintained in the population, it allows us to evolve and to adapt much quicker than organisms that have either less genetic variability or no genetic variability amongst individuals in their gene pool. Okay, but more on that a little bit later on in the semester. Okay, so as far as processes that are involved in, the term, sorry, I forgot about that. The term chiasmata. The chiasmata is the actual site in which crossing over occurs. Okay, that's just a term that they just have to know. So, as far as the actual processes of meiosis 1, here we have prophase 1, metaphase 1, anaphase 1, and telophase 1, PMAT, a good way to remember. So we do start off with our basic diploid cell coming in through the basic cell cycle. So we do undergo that basic cell cycle at the beginning, interphase, and then we move into prophase 1, okay, which here, instead of having the regular X chromosomes, we have the two X's forming our basic tetrad. And so this tetrad of chromosomes, which have now condensed, of course, into chromosomes, you have the breaking down of the nuclear envelopes, very similar things that we saw in prophase 1, in prophase during mitosis, only the difference here is the pairing of the homologs, okay, through the use of the synaptonemal complex. And also we have a crossing over, which occurs during prophase 1, along with all of the other things that we talked about during mitosis. Now in metaphase 1, we still have that basic concept as far as having all the cells uniting in the middle. All the chromosomes united in the middle of the cell, okay, in the metaphase plate. And from there, you do have the separation of the chromosomes, but in this case, you essentially have a separation of the entire homolog from one side, from one pole, and the other homolog over to the other pole. Okay, these are complete homologous chromosomes that are separated out, not simple, essentially sister chromatids that are pulled apart, like we see in mitosis. So here we have telophase 1, where we do have the separation of the two cells via the use of essentially cytokinesis at the end of that process by cleaving the cell into two. Okay, as we can see here very clearly, we have the reformation of the nuclear envelope, all the things again reforming that unformed during prophase 1. two cells that emerge from the original parental cell at the beginning. So between meiosis 1 and meiosis 2, we have interkinesis. And what interkinesis is it is a shortened version of interphase. So interkinesis, you have you don't have an S phase, so you don't have DNA replication occur, because that would be counterintuitive. Because we want to have this basic reduction division, you want to start off with the diploid cell and end with the haploid cell. So if you went through DNA replication at this point, you would essentially keep getting more and more chromosomes versus reducing it by half, as which is the point of meiosis. Okay, to have this reduction division, this cutting in half the number of chromosomes that we have in each individual cell, that later on, during the process of fertilization, we can have the paternal and the maternal homologs come together. In humans, it would be 23 and 23 chromosomes that come together to form our 23 pairs of 46 chromosomes that we think of as our typical diploid number in humans. Now, meiosis 2 is essentially the same basic processes that we see in mitosis. So here we see prophase 2, which you can basically consider the same processes as prophase during mitosis. Same concept with metaphase 2, anaphase 2, and telophase 2. Okay, the same concepts that we talked about in chapter 10 as far as mitosis, it happens exactly the same now here during the second meiotic division, which leads us to the formation. This is one of the major differences. At the end of the process, we actually end up with 4 haploid cells, 4 haploid daughter cells during meiosis, while in mitosis you actually end up with 2 diploid cells. Mitosis, 2 diploid cells, meiosis, 4 haploid cells. Okay? Now, one of the most important parts of the chapter is this last section, okay, section 11.4, which, as the title says, it basically sums up the major points between the differences between meiosis and mitosis. Okay, so it's something. So figure 11.9, which shows you the major differences, literally puts the two diagrams that we saw in this video and the video prior when we talked about mitosis. It puts them together so that they're able to easily compare the two. Okay, and that is basically the end of chapter 11. A lot of it is just, again, references back to the mitosis chapter. Okay, so this is the end of this particular workshop. If you have any questions, feel free and let me know.